Right. So, continuing our march through eukaryotic diversity, and today we should actually finish up with the protists. Here's where we are. I've said my piece about those diaphoreticales. On Tuesday, we had the alveolates, and then that group that, excuse me, may not be monophyletic, the excavata. And that leaves us with the amorphia, which includes two big groups, the Amoebozoa and the Opisthokonta. And once we get to the Opisthokonta, we'll finally be getting close to the actual animal kingdom after only, 3 weeks of lecture. I've said this before, but amoeba shape has evolved in multiple groups independently. Uh, uh, Great example of convergent evolution used to be that all amoebas were classified in the phylum Sarcodyna. And I only mention that because if you ever have to look at older references, you might see them called that. We don't use that term now. And the Amoebozoa, as we think of it now, is based primarily on similarities in DNA sequence because many amoebas are not in the Amoebozoa. Species of Amoebozoa will have a tendency towards a particular shape and size. There are some that are usually big, some that are usually small, some that usually are fan shaped, and some that are blobby, and so on. So you can actually tell amoeba species apart by looking to an extent. But cells have no fixed shape, and amoeboid locomotion involves the formation of at least one and sometimes multiple extensions of the cell called pseudopods, and the cytoplasm flows into the pseudopods, moving the entire amoeba forward. The secret to which is the fact that the cytoplasm can transform between a more flowing liquid state called the sol and a firmer, more jello like state called the gel. If you saw any live amoebas in that lab we attempted to do a week ago, I know at least two people got them. If you saw any, then you will have seen what I mean by amoeboid locomotion. Incidentally, we've seen the word test before in the context of foraminifera, which are way off in a group called the Rhizaria. Some members of the Amoebozoa also have shells or tests, and in this case, they tend to be a lot simpler than the more complicated multi-chambered tests of forams. These typically consist of only one chamber, and they can be made of secreted organic matter, sort of like chitin. They can be made of bits of silica. They can be made of calcium carbonate in one species, or they can be made of little mineral grains that have glumped together. That's a good word, glump. There's your classic large amoeba, which happens to be in the genus Amoeba, Amoeba proteus. This is the one that I ordered for the last lab that we attempted to do. You can see multiple pseudopodia. There's one, there's one, there's one, all of these little extensions of the cell. Two of the pseudopodia look like they're surrounding this little green alga right there. That's feeding by phagocytosis, the same feeding mechanism that you'd see in white blood cells, for example. So you've got multiple pseudopods in this case, and this is a pretty big cell. About 500 microns would be typical. And there is cell polarity to it. The pseudopods tend to come out of this end over here on the right. At the left, there's a semi-permanent kind of lumpy structure that trails behind the main direction of locomotion. It's called the uroid. Euro is Greek for tail. So your urologist treats diseases of your tail. Ah, ha, 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 ha. Uroid means it's shaped like a tail, and it is the part that tends to drag behind. And locomotion depends on sol gel transformation. And I talked about this in lab, but this diagram might make things a little bit more clear. The inner cytoplasm is in a more liquid state. That's the sol. And the sol is squeezed forward through the middle of a pseudopod. And then it kind of fountains around, pushing the cell membrane forward. And as it fountains around right here, um, subunits of the protein actin lock together, and the consistency of the cytoplasm becomes more jello-like, more solid. It's almost kind of like a tank tread where you have active movement up here, and then when we come around like this, the cytoplasm um, solidifies, those actin subunits lock together, and at the rear we have units of myosin that bind with the actin and contract to squeeze the endoplasm forward. So locomotion is made possible by actin and myosin, the same proteins that work in your muscle. They just operate in a very different way, very different arrangement. The amoeba that you saw earlier is pretty big, but there's also lots of small ones. And you can tell them apart surprisingly well sometimes by just looking for differences in shape. So at the top left, that's an acanthamoeba, very common species in moist soil. And acanthamoeba happens to have kind of cone-shaped pseudopods. Acantho means a thorn, so acanthamoeba looks kind of thorny. Flabellula at the top right only has a single pseudopod, and it tends to be very broad and fan-like. Philamoeba has very narrow and thin pseudopods. Thecamoeba has kind of a wrinkly exterior. And again, even though they don't have a fixed shape, they do have tendencies that you can key in on. One that the pre-meds among us may need to know, good old Entamoeba histolytica, happens to be the causative agent of amoebic dysentery. These amoebas get into your intestine. They can cause severe diarrhea. They can cause bleeding. They can erode the lining of your intestine. And in really extreme cases, they can perforate your intestine and escape into all sorts of places you really don't want them. They can create these enormous abscesses in the liver, and they can, in fact, kill you. And wherever you are when you hear these words, please take off your hat and remember the German microbiologist Felix Schauden, who worked out the life cycle of Amoeba histolytica by deliberately infecting himself with Entamoeba histolytica, and in fact, it went on to kill him. So, yeah, the history of parasitology has a number of people who tested infective agents on themselves, sometimes at the cost of their lives. Anyway, Entamoeba histolytica, there's an active feeding stage at the left. That's called a trophozoite. Trophane is again the Greek word for to feed. So trophozoite just means the feeding stage. And those alternate between trophozoites and inactive stages that have a thick wall around themselves called cysts. And it's the cysts that pass out in the feces and get onto things like contaminated food or water that the next person feeds on. This is kind of gross, but this means of transmission in microbiology and parasitology is called the fecal oral route. The cysts pass out in the feces, and if you don't have good control of sanitation, if you're in a place where, for example, Farm workers aren't given restrooms to use, or human waste is used as fertilizer, or people are just not real careful about washing their hands. The cysts can get onto food or can get in water that others will ingest, and that's how this thing passes. That's how this thing goes from one person to the next. There's a few others that can cause disease. That's acanthamoeba. And again, it's one of the most abundant protists in soil. At the left, there's a trophozoite. You can see that it's got little spiny looking pseudopods. On the right, that is an acanthamoeba cyst. And these can dry out and these can survive for months, possibly years, waiting for water to return and conditions to come back. Acanthamoeba is not very big at all, and the cysts are not very big, and the cysts are very easily spread in dust. And if they get into your eyes, they can infect your cornea. That's an eye with a serious acanthamoeba infection going on. This could cause permanent blindness. This guy may well need a corneal transplant. And acanthamoeba is not really a full-time parasite, and it's not normally spread from person to person, but it can cause these sort of accidental human infections. This is the kind of thing you're at risk for if you don't use sterile saline on your contact lenses and you don't change them regularly. You can end up with some pretty nasty stuff on your cornea. Acanthamoeba is also packed full of bacterial symbionts, and we don't entirely know what these are doing, but some of these symbionts can leave the cell and can cause human disease. The bacterium Legionella causes Legionnaire's disease. That's a very severe lung infection. It's called that because it struck at a meeting in a hotel in Philadelphia of members of a veterans group called the American Legion. And they suddenly started coming down with terrible lung infections, and several of them died because Legionella was hanging out in water in the air conditioning system's cooling towers. But Legionella can also be spread and can pass from one environment to the other, hanging out inside acanthamoeba. This is another acanthamoeba packed full of a different respiratory pathogen called parachlamydia. If you've ever hung out with Dr. Hazelcorn, you might know that there's a number of amoebas that have bacterial symbionts that can sort of hitchhike inside the cytoplasm of the amoeba itself. Exactly what they're doing, well, that's one of the things that she's interested in. I don't think we fully understand that as of yet. Testate amoebas are quite common in freshwater environments, and these are completely harmless. This one's called Arcella, and you're seeing a top view. This brown thing that you're seeing right there is the test, and in 3 dimensions, this thing would look kind of like a big floppy beret. This round thing right there, that's the opening. That's where the head would go. Most of the amoeba cell itself is inside the test. But there's this opening in the bottom, and coming out of the bottom, you have multiple pseudopods. Okay, one moment. I have just gotten a. Oh, guess what? We are still on remote instruction tomorrow. Just now got the word. I'm sure you will soon if you haven't already. Okay, well, I'll have to come up with lab replacements and I'll just do it at the last minute and I'm sure it'll be fine. That's always works, right? So that's a testate amoeba. There's a few more. These are 3 different species of the test of an amoeba called Diflugia. The test is made from sediment grains that have been glued together in a pretty regular fashion. In other species, the cell might be made of particles made by the amoeba itself. And in the case of Arcella, that's just some kind of tough organic matter. I don't absolutely know if it's truly chitin, but it may be. We now know that the Amoebozoa also includes a lot of critters called slime molds, which used to be classified with the fungi. Physarum is an acellular slime mold. It's basically a giant amoeba that can form a actively growing stage of, I don't know how big the biggest one is, but this thing can span several inches, maybe a foot or more. This is basically one giant cell with many thousands of nuclei. You find these in moist terrestrial habitats, in places like very wet forests. You might find these covering a sizable portion of the underside of a log, for example. So they can get to be quite large. There's a lot of cytoplasmic streaming that's constantly going on, transporting nutrients and everything else. And when conditions are bad, they can form resistant stages and just wait it out. They also include cellular slime molds. Dictyostelium is really cool because Dictyostelium spends most of its life as a small free living amoebas, crawling around in damp leaf litter and damp soil and damp vegetation, feeding on bacteria and living its best amoeba life. But when Dictyostelium is stressed, they come together and they aggregate into these millimeter sized stages called slugs. There's a slug right there at the lower left, and the slug will just crawl around until it starts kind of humping up, and then it starts elongating and it forms this structure. The order of stage appearance, you're basically going from this to that, and then the slug kind of, yeah, here the slug stops moving, lumps up, forms this kind of hat stage, and then forms a stalk that elongates and elongates. And the amoebas that got to the top of the stalk, the ones up here, form cell walls and become spores. And when the whole thing dries out, the spores will drift away on the wind. And if the spores make it somewhere, they will end up, if they make it to a place with good conditions, they will hatch and end up as free living amoebas again. So this is a critter that is multicellular some of the time, but spend most of its time as a single-celled organism, which is a pretty darn nifty trick. And these are the amoebas that Dr. Hazelcorn studies in her own lab. It's a really interesting evolutionary question because this is a great case of altruism, because the stalk, all of this, is also made up of amoebas. The amoebas form that stalk. They make a cell wall to make the stalk firm, but they die. The amoebas that make the stalk are essentially sacrificing their lives for the sake of the amoebas up in the sporangium. And how that could evolve is actually a really interesting question that I don't think we've fully solved. It's an example of what's called evolutionary altruism, and it's a really cool area of research. So much for the amoebozoa. That's all I get to say about amoebas. The last eukaryote group that we have to cover is the Opisthokonta, and this is the group that includes the fungi and it includes the animals. Opisthokonta means kanta, I think, is a whip, and opisthe is the Greek root for to the rear. So Opisthokonta, during at least one stage in their life cycle, have a single posterior flagellum, or at least we think their ancestors did. Um, a lot of opisthokonts, notably most of the fungi, have completely ditched the flagellum. There's one group called the chytrids that still does retain it, but all of the mushrooms that you're likely to know, for example, never have a flagellated stage at all. But there are still protist opisthokonts that do. Certainly in mammals and in most animals, sperm have a single posterior flagellum, although that's not universal. And again, the main opisthokonts are the fungi and the animals, and there are some single-celled protists that are more closely related to either of those than they are to different protist groups. So this is a funny little amoeba called Nuclearia. Looks a little bit like a fried egg, except it's got these really long pseudopods. Despite its amoeboid shape, we don't put it in the Amoebozoa. DNA sequencing suggests that this is, in fact, an opisthokonta, and it's closer to the fungi than to anything else. So that is an amoeba that is not in the Amoebozoa. I'm not going to say very much about fungi in this class. Um, wish we had a mycology class on the books, but we haven't had anybody here that could teach it for maybe the past 15 years. But true fungi include yeasts, they include bread mold and athlete's foot fungi and bracket fungi and jelly fungi and these hallucinogenic Amanita muscaria mushroom fungi. Very prominent Opisthokonta clade. And purely because I can, there are lots of mushrooms in the nature reserve starting out in spring. In a couple of months, things are going to get pretty mushroomy there. And both of these are taken in the nature reserve. To the left, that is an unknown species of bolete mushroom. The underside doesn't have gills. It's got this kind of spongy layer. That's what makes this a bolete. And boletes are often pretty brightly colored. The underside of this cap is brilliant yellow. On the right, that is a bolete mushroom, probably the same species, but that one has been parasitized by a bolete-eating fungus called Hypomyces chrysospermus. So all of that whitish film that you see covering that mushroom, that is Hypomyces chrysospermus growing on top of the mushroom and slowly digesting it alive. So this is kind of like uh, yeah, this is kind of like a zombie mushroom, if you will. And yeah, both of those I found in the Jewel Moore Nature Reserve a couple of years ago. Wonderful stuff. I love it. There are fungi that cause human disease. Healthy people can get things like athlete's foot and ringworm and barber's itch and tinea versicolor. Fungal diseases of the skin are pretty common. Fungal diseases deep in the body are they're rarer, but they're extremely dangerous, especially to people who are immunocompromised. And one group of fungi called Microsporidia used to be considered parasitic protists, but DNA sequencing has shown that what they actually are is extremely stripped down fungi. They don't grow in filaments. They're essentially single celled, and they've got some of the smallest genomes of any eukaryote, up to 1,000 times smaller than your own genome. There's, I think, 8 that have been found causing human infections. Most of them are pretty rare, not the sort of thing you'd see very often. But this one, Enterocytozoon, causes diarrhea and wasting in immunocompromised patients. So this is the kind of thing that those of you going on to medical school might well someday see. And that's all the fungi we got time for. Except I can always tell my joke as to why did everybody love the mushroom. The answer is, of course, he was a fun guy. And I'm just imagining everybody laughing uproariously right now. Anyway, all opisthokonts that are closer to Animalia than to fungi are sometimes grouped in the holozoa. This is one. This is a very weird little amoeba thing called Capsospora of Charzaki. Very small, lives, if memory serves, in the blood system of an African land snail. Exactly what it's doing there, I'm not entirely sure. What we do know from sequencing is its DNA is that it is closely related to the animals, even though it is not itself an animal. And it's got some molecular features that only animals, well, we thought only animals had. Notably, it's got a family of transmembrane proteins called integrins. Integrins cross the plasma membrane. There's a part sticking out and a part hanging in to the cell. In animals, integrins bind to the extracellular matrix. One of the distinguishing features of true animals is that their cells secrete fibers outside of the cell itself, fibers of collagen and a bunch of other things, proteins and glycoproteins and such. That makes up what we call the extracellular matrix, this network, this meshwork of proteins that our cells are held to. And in animals, integrins bind to the extracellular matrix, so they help hold an animal body together. That internal part of an integrin protein can also, well, integrins do signal transduction. That internal part of an integrin protein can trigger a change inside a cell. And so animals use these for things like cell cycle uh, cell cycle regulation. They're a major part of the way that animal cells can sense and respond to what's going on around them. And they're not unique to animals. It turns out that holozoans have these too. So this thing has integrin proteins, even though it is not multicellular. The same is true for Ministeria vibrans. It's another weird little amoeba that nobody has paid much attention to, but we know that it can form clusters when certain chemical cues are present, and we know it's got integrin genes as well. So this thing, although it's not fully in the animal kingdom, it has at least some of the molecular machinery found otherwise in animal cells, which is a pretty evolutionarily significant thing. Suggests that a lot of the machinery needed to make a multicellular body evolved for some other reason before the evolution of multicellular bodies, and then got reused. And exactly how that happens, you figure it out, and we can write a paper on it. So animals are made of more than one cell, but there are lots of non-animals that are too. And I'm not even talking plants and fungi. There are plenty of eukaryotes that form multicellular colonies. These happen to be diatoms. These are in the straminopiles. Those are in that S group in SAR. They're not closely related to plants or animals at all. But you can see they make fairly complex multicellular colonies. Um, Other straminopiles can make even larger and more complicated bodies. I'm thinking of things like brown seaweeds and those can get quite large and can get fairly complex. So it's not only multicellularity that's unique to animals. Um, what makes an animal is, to an extent, cell differentiation. It's not just that animals have many cells, but they have many different cell types, anywhere from four in the simplest of animals to roughly 200 in you. Your body's made of roughly 200 different types of cell, whereas the simplest animals that we're going to see have exactly four types of cell. But you have to have cell differentiation to be a true animal. Although, be it said, plants have this, fungi have this, and some of the more complex things like seaweeds have this too. So this is not unique to Animalia. What does seem to be unique is that animal cells are held together um, by an extracellular matrix, a network of mostly protein and glycoprotein fibers, notably the protein collagen, plus other proteins with names like elastin, fibronectin, and laminin, and various others. There's actually multiple types of collagen. There's something like 30, um, and different types are used in different tissues in different conditions. But all animals are going to have their cells held together with an extracellular matrix. The extracellular matrix also contains proteoglycans, which are proteins with carbohydrates stuck on. One, excuse me, one proteoglycan that you might have heard of that's found in cartilage is chondroitin. And I don't know if this is marketed to you in particular, but chondroitin supplements are being marketed as dietary supplements that supposedly help people with arthritis. I've never taken the stuff myself. I have no idea um, whether they work or not, but that is a proteoglycan that you might have seen. Animal cells have cell adhesion molecules, sometimes called CAMs, which hold cells to each other, and substrate adhesion molecules, or SAMs. The most common class of cell adhesion molecules are called cadherins, and the most common class of substrate adhesion molecules are the integrins, which I've mentioned earlier. These enable cells to hook together, and they're not just physical linkages, they're important in signal transduction as well. And finally, animal cells can be held together by tight junctions. They can also be held together by gap junctions, which allow ions to pass from one cell to another and allow cells to be electrically coupled. And we'll see these. This happens to be some of the tissue that you might find under the skin of your cheeks. It's called loose areolar connective tissue. The cells are, summing up the laser pointer, each one of these dots right here is a cell. And then all of this fibrous, stringy stuff are fibers of collagen and fibers of elastin that hold the cells together and give the entire tissue its soft and springy nature. And this is just a nice way to see the extracellular matrix, or the ECM. Not all human tissues have nearly this much. You can, I'm not recommending it, but you can get injections of collagen under your skin to add more of these fibers and kind of plump up your extracellular matrix. So some people get this to get thicker and fuller lips. Um, I don't know if people get this injected into their butts, but I guess you could. Anywhere the body needs a bit of plumping up and fulling out, you can add extra collagen. Not saying I recommend it, but it's an option these days. These are epithelial cells. Not sure from what part of the body, although they look pretty close to what you would find on the inside of your mouth. And these have been imaged with a confocal microscope. You can see the nuclei are staining blue, the cadherin proteins are staining red, and these are the proteins that hold one cell to another cell. They form these cell to cell junctions. So you can see there's a lot of them right where each cell meets every other cell. Gap junctions join one cell to the other. They're made of these protein subunits that can open or close, and they can allow ions and small molecules to pass from one cell to the next. And these allow electrical coupling. These mean that electrical signals, like action potentials, can easily pass from one cell to the next. So you get a lot of these in cardiac muscle. Heart muscle cells pick up a stimulus, um, ultimately from a little lump of modified cells called the sinoatrial node. And heart muscle cells pass along this stimulus in a very particular pattern, which is what enables the heart to beat in the very precise way that it does. So that's made possible with gap junctions. I always thought a gap junction would be like a store at the intersection of two roads where you could buy casual fashion, would be a gap junction, but I guess not. I'm imagining all of you laughing hysterically. Okay, integrins look like that. You see that they're transmembrane proteins. The membrane is just kind of shown here as a gray band, and they can bind to the extracellular matrix. You can see one on the left that is not, one on the right, that little dash line there, that would be a fiber of maybe collagen. You can see that binding has caused the entire thing to change shape. And these integrins activate signal pathways within the cell. So this part that's sticking inside will change shape, and it might be attached to the cell's cytoskeleton. And that shape change might cause the cell to start moving in a particular direction. Um, that change might trigger the release of cyclic adenosine monophosphate or something like that. There's lots of different ways that a conformational change of the integrin protein down here will activate a signaling pathway within the cell. So the cell can respond to the matrix that it has adhered to, which turns out to be really important in development. That's probably a zebrafish embryo right there. But there was a time when you didn't look so terribly different. You have, that's going to be the eye, that's the nasal placode right there. These clefts right here, the pharyngeal clefts, you had them at one stage. In this zebrafish, they're going to become gills, or gill arches anyway. And right at this stage, right when the central nervous system is at its earliest stages, there's a population of cells that buds off of the developing tube-like central nervous system. They bud off of a structure called the neural crest, so they're called, excuse me, neural crest cells. And the neural crest cells migrate away from the newly forming central nervous system, and they go on to form key structures in the head and the face. If your neural crest cells don't migrate properly when you're an embryo, you can end up with some pretty horrible facial deformities. Neural crest cells also migrate through the body and give rise to ganglia, to, nerve centers outside of the central nervous system. And that's one of the key features that vertebrates have. It's very important for forming things like faces. And neural crest cells, quote, know where to go because their integrins bind specifically to an extracellular matrix protein called fibronectin. So this is key to how the neural crest cells, quote, know how and where to migrate and probably would have a role in what these neural crest cells transform into as well. So that is also, I mean, neural crest cells are only found in the vertebrates, but the overall idea of cells in a multicelled body binding to extracellular matrix and navigating that way, that's something you'll find throughout the animal kingdom. And as we've seen, those integrins make it possible in part for animals to develop from embryos. On the gene side, animals have whole families of genes that are called homeotic genes. And homeotic genes are arranged in a hierarchy. What you're looking at here up at A is a fruit fly embryo that is expressing one gene at the anterior end. I think this one is called bicoid, and a second gene, which I think is called crouple, uh, in the middle. And these different domains of bicoid and crouple have been stained green and red, respectively. At a later stage, bicoid and crouple and some others we're not seeing, like caudal, switch on the expression of other genes, seen here in B. This is a segmentation gene. I think it's called Fushitarazu. And those will turn on a whole nother set of genes that lay down more of the segmentation pattern. You see that in C. And then those will turn on a set of genes that are found only in the animal kingdom called Hox genes. And Hox genes govern segment identity. Hox genes essentially tell a developing embryo where you're going to put the legs, where you're going to put the eyes, um, where you're going to put the head, where you're going to put the butt. Um, they're responsible for laying down a lot of the basic body plan. And these are fruit fly embryos, but you've got Hox genes. Every animal has Hox genes. Even sponges have a few. You've got multiple clusters of them. And this ability to specify different regions of a developing embryo by having whole batteries of genes at one level that promote and repress sets of genes at a lower level is basically how you build an embryo and ultimately an adult from a single fertilized egg. So animals have these families of homeotic genes, where families at one level promote and repress families at another level, and collectively they lay down the basic body pattern very early in development. You have all of these genes. Roundworms have all these sets of genes, even though their bodies don't have any segments. It's a universal toolkit for building bodies in the kingdom Animalia. Okay, we're not quite with the animals yet. The closest living protists to Animalia are a group called the choanoflagellates. And like some of those other weird protists that we've seen, they actually do contain cadherins. Sorry, I think they contain integrins, but they also contain cadherins, which are cell-to-cell adhesion molecules. They also have these surface receptors that are critical for cell-to-cell signaling, called receptor tyrosine kinases. So these have even more of the molecular machinery that true animals do. And in fact, we now know that many choanoflagellates can form multi-celled clusters. They're not necessarily permanent clusters, but they have a lot of the molecules that you need for taking a bunch of cells and clumping them together and making a larger integrated unit. Choanoflagellates tend to be really small. I don't know that I've ever seen a living one because, here I don't usually use oil immersion microscopes, and you'd really need that to be able to spot a choanoflagellate. They do have one single flagellum, and the base of the flagellum is surrounded by a ring of microvilli, a ring of more or less permanent finger-like projections around the base of the flagellum makes up a structure called the collar, because it evidently looked to an early microscopist like a collar, like a shirt collar. Excuse me. Soon be time for me to wrap up in these blankets and go back to bed, but I got to finish this. How are we doing on time? Loads of time. Colossal amounts of time. As the flagellum beats, water gets pulled through the ring of microvilli, and the microvilli will sieve out tiny food particles, which the cell can then phagocytose. So that's how they feed. And there's a diagram of what I mean. The cell body is usually just kind of oval and it's not shown, but there's a nucleus in there. You have that ring of these little finger like projections, the microvilli. And in the center of the ring, you've got the root of a long flagellum. And as the flagellum beats back and forth, it pulls water. And I want to draw this. It pulls water that way. So water passes through the microvilli, between the microvilli, and then upwards and backwards, because the cell is swimming this way. Don't know why we got that extra line in there, but there's a forward arrow. So the water gets shoved backwards, and as it passes through the microvilli can sieve out tiny little food particles and phagocytose them and feed on them that way. That's one that's only 6 microns in size, kind of shaped like a carrot. And there's the cell body. That there's the flagellum. And then this right here, that's that collar. And the microvilli are so close together that you can't resolve them separately under the light microscope. They just look like a continuous structure. There's another one, Monocyga, on the left. Monocyga has a stalk. Some choanoflagellates are sessile. They'll use a stalk to attach themselves to something and just stay there. And you can see the flagellums in the middle right there. And then those are, you're looking at a cross section through the collar. So that and that are parts of the collar. Whoops. Salpingoica on the right, very similar genus. You can see the stalk, you can see the flagellum, and now you can see how those microvilli make up that collar, make up that ring of microvilli. There are a bunch of marine species that secrete little silica bars, which they can weave together to make a basket-like structure called a lorica. Lorica is a term you'll see again. It's Latin for a breastplate in armor. If you've ever seen movies about ancient Rome, where you have the centurions wearing this big chest plate made out of bronze that's usually sculpted so that it's got an absolutely amazing set of pecs and a big six pack. That's a lorica, or at least that's one type of lorica. A lorica is a breastplate, and you'll see it used in some other groups, not just this one, for a protective structure that reminded the guy who described it of a breastplate, quite possibly because he was on drugs. Anyway, these are marine choanoflagellates that secrete these silica bars that stick together to make up this lorica. Where things get weird is that some choanoflagellates are actually colonial. Sometimes this is permanent. In other species, they may go through temporary colonial phases during the life cycle. In some cases, they're single most of the time, but changes in water chemistry will trigger them to start clumping up. So choanoflagellates don't just have these cadherins and tyrosine kinases and things like that. They use them to make multicellular stages, or at least sometimes transitory multicellular stages. In some of these colonies, like these of Salpingoica, the cells don't just clump up, they communicate with each other and they synchronize their behavior. And they synchronize their behavior using these calcium waves, the same sort of mechanism that governs the locomotion of ciliates, which we tried to demonstrate in the first lab that we did. There's a recently described one called Baroica that forms hollow colonies. There's a space outside and an inside. Apparently, these have some sort of extracellular matrix holding the cells together, so that's very animal-like. And apparently, there's a whole microbiome inside Baroica colonies. This was only just described a couple of years ago. There's a Dr. King over at Berkeley has really led the way of looking really hard at choanoflagellates. Her lab has documented a whole bunch of new species and has described all sorts of crazy things that they do. And I think they were the ones that described Baroica. And a very rare one called Proterospungia, which hasn't been seen terribly often, is not just colonial and doesn't just have an extracellular matrix, but it's got cell differentiation. The flagellated cells are on the outside and enable the thing to swim. On the inside, you have amoeba-like cells, which apparently can transform into other types of cell if the colony needs to regenerate part of itself. So this is all really, really, really sounding animal-like, and I hope you see that choanoflagellates collectively range from very protist-like to very, very close to all of the features that animals would have. This is knocking right on the door of the animal kingdom. And the reason that matters is that choanoflagellates like this one, seen here using scanning electron microscopy, there's one of these colonies, and you can see the flagella. There we go. With the microvilli are dead ringers for choanocyte cells, which are found inside the simplest and maybe, although maybe not, although we're not really sure, but possibly the earliest branch of the animal kingdom, the what we used to call the porifera, the sponges. Ta-da! We have finally reached the animal kingdom. In the time that we've got, I'm going to start introducing the phylum Porifera. I'm not going to finish all of it. We used to group all sponges into one phylum. We now consider that's a paraphyletic group, and I'll talk later as to how we classify sponges today. Um, how much do I want to get into this right now? Probably I'll wait until next week. It's still okay to talk about porifera informally but we don't think they represent a monophyletic taxon anymore. So it's okay to talk about sponges if you recognize that they don't, sponges are no longer considered to be all and only the descendants of one particular common ancestor. Um, Although maybe they are, because there's actually been a 25-year debate brewing as to exactly what the very base of the animal kingdom looks like, and I'll get into that after I've covered some more detail and introduce you to the deadly struggle between the poriferan model and the ctenophore model. But we'll get to that later. There are a few sponges in freshwater, but the great majority of these are marine. There's about 5,000 species known in every marine habitat from tide pools all the way down to the abyssal zone, 29,000 feet below the surface. They're all benthic. They live on the bottom, and they're all more or less permanently attached to a substrate. That would be a rock or a shell, some kind of solid object. For most of them, if they attach, it's permanent. There's at least one species, the chicken liver sponge, that has been observed in a lab attaching to aquarium glass and shifting its position by as much as an absolutely whopping 4 millimeters per day. So these sponges, a few sponges can in fact move, but 4 millimeters per day, they are not going to win Olympic gold medals anytime soon. The fastest sponge of all was a chicken liver sponge that in a lab where I assume the grad students were the ones that nicknamed it Speedy Gonzales because of its lightning fast 4 millimeter a day motion. There's a few sponges that are carnivores, but classic sponges are filter feeders, and the entire body is one great big water filter. Water gets pumped into small holes called ostia, and where it can go from there depends on how complex the sponge is. But eventually, that filtered water gets shot out of the sponge through one or more larger holes called oscula. So, water is taken in through the ostia, these little holes. Food particles are filtered out, and the filtered water passes out through larger holes called oscula. Within the sponge, food particles get filtered out by choanocytes. These are the so-called collar cells that look exactly like choanoflagellate cells. They both have a collar of microvilli, and the choanoflagellates will phagocytose little food particles and then pass those on to other cells. So there's a view of some sponge tissue. These are a couple of choanocytes from a sponge, and there's no difference worth mentioning between sponge choanocytes and free-living choanoflagellates. You can see you've got this kind of globe-shaped cell body and a collar of microvilli. And the microvilli are coated with a sticky layer of snot called the glycocalyx. The glycocalyx is what's making these little cross attachments between the microvilli. So the glycocalyx kind of webs the microvilli together, and it improves the ability to trap particles. Please excuse me. Sorry, I sound like I'm on the verge of running out of sleep, but we ran out of coffee yesterday. I sure hope we get the driveway cleared. This is getting bad, folks. Right. There's other types of cells in a sponge as well. Pinacocytes are flattened cells that form an outer layer, an outer quote skin, although it isn't really the same as the skin of more complex animals. The choanocytes you've just seen, those pump and filter water. Choanocytes, when they phagocytose, will pass on food particles to these amoeba-like cells called archaeocytes. Archaeocytes will just wander through the body and they can differentiate into any other cell type that's needed. So if there's damage to a sponge, the archaeocytes can come wandering to the damaged spot and transform into other cells, which enables the sponge to regenerate. And they can do this quite well. Sponges are generally very good at regeneration. Archaeocytes can also pass food particles to other types of cell. So they can pass nutrients to other cells that are not feeding on their own. Specialized cells called the sclerocytes secrete the elements of a sponge skeleton. Not all sponges have this, but many of them have skeletons made up of nearly microscopic little bits of either silica or calcium carbonate that can look like little needles or can look like little stars that can actually take quite a large number of shapes. But all of those little sharp bitskis of silica or calcium carbonate are collectively called spicules. And sclerocytes secrete new spicules. These may lock together to form a support structure for the entire sponge. They're also a pretty good anti-predator defense because most predators don't want to take a bite into something that's basically a heap of microscopic glass shards. Whoops. Sponges are also supported by collagen fibers secreted by special cells called collocytes. Some sponges secrete a different tough protein called spongin, which would be secreted by lophocytes. Spongin, by the way, there are some sponges that don't produce spicules, and they're entirely supported by fibers of spongin proteins. These are the natural bath sponges. You used to be able to buy these at Bed Bath and Beyond, or maybe it was Bath and Body Works, or whatever the store that went broke a couple of years ago. Spongin is very tough, but it's also very flexible, and spongin fibers forming this very dense, spongy network. Um, well, that's basically what a natural bath sponge are. Natural sponges are still harvested from the bottom of the ocean and can be used for scrubbing. You'll see some samples when we get to it in next week's lab. The entire body can be capable of contraction, although it's very slow. There are myocytes that can cause the body to contract. Porocytes would surround the ostia and the oscula, and those can either expand or contract to open or close the pores. And then finally, sponges reproduce sexually, producing egg cells and sperm cells. Sponges do not have true tissues. A true tissue has cells that are connected by gap junctions. And in a true tissue, usually you have a sheet of cells that are all bonded to a very thin membrane of extracellular collagen fibers called the basal lamina or basement membrane. Sponges don't have these except for the larvae of one group of sponges called homoscleromorphs that actually do show true tissue. And this group, the homoscleromorphs, seem to be more closely related to the rest of the animal kingdom than other sponges are, which is what makes the sponges paraphyletic. Again, more on that when we reconvene. So sponges don't have true tissues. They're just cells that are kind of hanging around and hanging out in formation. The outer surfaces are lined by pinacocytes. Pinax happens to be Greek for a flat wooden tablet. So pinacocytes mean cells that are thin and flat, and they butt their edges up together and they make a skin-like surface called the panacoderm. The inner surfaces are lined by a layer of choanocytes, that's the choanoderm, and the space between is filled with a collagenous mass known as the mesohyal. Hyle happens to be the Greek word just for matter. So the mesohyal is the middle matter, and the archaeocytes crawl through the mesohyal. Spicules are embedded in the mesohyal. The sclerocytes that make the spicules do their work in the mesohyal. Um, all of that stuff takes place in the mesohyal. So your basic sponge is going to have, sorry, I did not mean to expand that. There. Over on the left, we have the choanoderm, which is just a whole bunch of choanocyte cells, all with their flagella, all beating. Over on the right, these flat cells, you're seeing them edge on. In life, they would kind of butt together like tiles. Those are the panacocytes, and the outer layer is the panacoderm. Here we've got one of these tiny little incurrent pores, an ostium, and holding the ostium open is one of these porocytes, and the porocytes can open or close to increase or decrease the flow of water into the sponge. And then all the gray stuff in between, that's the mesohyal. There's an egg cell developing in the mesohyal. Here we've got a lophocyte secreting fibers. And right here is one of these archaeocytes that can pick up phagocytized food particles from the choanocytes and pass them to other cells in the body. So archaeocytes do a lot of the work that in you would be done by the circulatory system. They are transporters of food. Okay, let me see. Do I want to go farther? Okay, I seem to be stuck there, but that's okay because it is just about time to wrap this up. When we resume lecture, hopefully face to face, I'll talk about how this basic kind of sandwich organization of a sponge body can be folded up in increasingly complicated ways. Sponges have much more sophisticated anatomy than you might think, and we will talk about that when we reconvene. But once I stop the recording, we can all cuss if we need to. So I'm stopping the recording now.